Hello students. So here the MCQ is, what will be the mRNA transcript if the coding strand of DNA is 5' ACCATG-3'. So these type of questions are frequently asked in the competitive exams, mainly the NEET UG and NEET PG in the medical branch. So here are the options: A, this is option B, and option C, and option D. Okay. So in this type of question, you have to understand the concept. Okay. So in DNA, it is a double-stranded DNA. The one strand is called as a template, another called as a non-template strand. Now, this template strand, okay, that is transcribed. Suppose this is a 3' end and this is a 5' end. So that will be transcribed to mRNA, okay, in the 5' to 3'. Okay, and this 3 to 5', that is called template strand, and the other strand of the DNA, that is will be 5' to 3'. Okay, here RNA polymerase read from 3 to 5' and polymerization takes place in 5' to 3'. So mRNA will be synthesized from 5' to 3'. This non-template strand has similar sequence to the mRNA strand. You can see here. Okay, and this is called as the coding strand, right? Which is also called as sense strand. While the template strand, that is called as a non-coding strand, and it is a anti-sense strand. So in this type of question, you have to understand the concept. Here, the coding strand of DNA is 5' to 3' is given. So here, this non-template strand is given from 5' to 3'. So you have to understand that mRNA will be also in the 5' to 3'. So, and except here the difference between the coding strand and mRNA is just in DNA there is a thymine. In DNA there will be thymine, while in RNA it will be uracil, right? So the difference will be similar to exactly here the T will be replaced by U. So if you see here 5' to 3', option A, you can see ACCATG. So it is a similar, so it will not be there because in mRNA there will be uracil. Fine. This option B, that 3' GUACCA. But if you read it from 5' to 3', that is A, C, C, A, T is replaced by U and G. So this is our correct answer, right? So always remember students, in exam you have to see which strand is asked. One, whether coding strand, whether template strand. If it is template strand is given, and you have to see the sequence, whether it is 3' to 5' or it is 5' to 3'. Okay, if 3' to 5' is given, then you have to just add the sequence from 5' to 3'. Okay, if template strand is given as a 5' to 3', you have to convert it to the 3' to 5', then add the sequence from 5' to 3', because mRNA transcript will always be in the 5' to 3'. Fine. So accordingly you have to see the sequence, what is that, right? So I hope it is clear to you. Thank you for watching. If you have any doubt, please write in the chat box, in the comment, and you can visit my website www.livebiochemistry.com. Thank you for watching.